Hello students, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to talk about a module that is enzyme in bacterial drug resistance. This has been taken from paper Molecular Enzymology and Protein Engineering. So objective of this module is to understand the role of enzymes or protein in bacterial drug resistance. So bacteria, as you know, they are pathogenic. They cause a number of diseases and antibiotics are used to control the diseases caused by these pathogens. But bacteria develop resistance against these pathogens. In this module, we have taken a beta-lactam drug which inhibits the cell wall biosynthesis as a model, and we will talk about the enzymes of the protein which is involved in the drug resistance of this antibiotic. So this module is divided into following sections. The first, we will introduce about bacterial drug resistance mechanism. Then we will discuss about different enzymes which are involved in the drug resistance, and then we will discuss what is the future prospects of this enzyme in the drug resistance. As you know, the pathogenic bacteria causes a number of diseases that are linked to high degree of mortality and morbidity. One such groups of pathogen is known as escape pathogen. ESKAPE, escape pathogen. They causes pneumonia, urinary tract infections, respiratory infections and other diseases. The important thing about this bacteria or these pathogen that they can grow on the artificial surfaces which is present in the hospital setup. They can utilize ethanol as a carbon source. This ethanol is used for the cleaning of hospital setup. They can resist the desiccation. They can survive at various temperature and pH condition, which makes these pathogen, escape pathogen, a lethal pathogen. So prevalence of these pathogenic gram-negative bacteria is in the clinical setup has increased gradually. Different antibiotics are used to treat this microorganism, which include beta-lactam, quinols, aminoglycoside, etc. But they have developed resistance against these antibiotics. So the resistance against these antibiotics has emerged in the microorganism, which creates significant health problem and responsible for high morbidity and mortality. And this make resistance mechanism or resistance strain is one of the major health concern. There are three main ways by which a pathogen acquires the resistance. It may be natural, it may be from lateral gene transfer, or it may be because of misuse of antibiotics. Natural resistance results from genetic mutation of the genes involved in the normal action of the antibiotics. A species may acquire resistance from other bacteria by lateral gene transfer via its plasmid or by other mechanism. The misuse or excessive use of antibiotics will also lead to the resistance in the pathogens. But out of these three, natural resistance, lateral transfer and misuse of the antibiotics, last two, that is species acquiring resistance by lateral gene transfer and misuse of the antibiotics, are most important for resistance development. The microorganisms that are resistant to different antibiotics, not one, they are known as multi-drug resistance or MDR strain or superbugs. Therefore, it is important to discuss the different resistance mechanism involved to control the antibiotics, or you can say to outcome the effect of antibiotics, and different proteins or enzyme which is involved in the bacterial drug resistance. As I told you, here we have taken beta-lactam as a model antibiotics, and we will discuss different resistance mechanism against this beta-lactam. different resistance mechanism. Different antibiotics are inactivated by four principal mechanism of resistance. In the present module, we have discussed the resistance mechanism against beta-lactam. So beta-lactams are antibiotics which inhibit the cell wall biosynthesis by inhibiting the penicillin binding protein, which is involved in the cross-linking of NAG and NAM, that is unit of cell wall peptidoglycan. And they are divided into penicillin group, cephalosporin group, monobactam group and carbapenem. Carbapenem are the recent most effective antibiotics. The four mechanism which is used to encounter beta-lactam, the first mechanism is alteration of antibiotic target site. This is now an important resistance mechanism for several pathogens such as gram-positive staphylococcus and streptococcus species. Here I am explaining about the general mechanism, and in the next slides we will discuss about the details of beta-lactam only. So the first mechanism is a common one. It is the alteration of antibiotic target site. The second mechanism is presence of efflux pump, which efflux antibiotic outside, and max ABOPRM efflux pump is a major cause of resistance in Pseudomonas and other pathogenic gram-negative bacteria. The third and most common mechanism is the production of enzyme that degrade or modify the antibiotics before it can reach the appropriate target site. The beta-lactam mase family of the enzyme degrade beta-lactam antibiotics and found to be widely discriminated among gram-positive and gram-negative. The fourth mechanism is alteration in the outer membrane protein, which is reported in Acinetobacter baumannii. Here you can see that we have a plasma membrane, or you can say the bacterial membrane, which is outer membrane, inner membrane and periplasmic space in between. So bacteria have outer membrane which have some channel and pumps which allows antibiotics to enter into the pathogen. So you can see that the beta-lactam antibiotics entering through a channel. So if the pathogen is alter this channel, the entry of beta-lactam is inhibited. The second you can see here, because we have taken the beta-lactam as a model, so the penicillin binding protein or PVPs is a target enzyme. So you can see that the PVPs are present in the inner membrane. So if the PVP get altered, the beta-lactam antibiotics which is enter into the pathogen will not bind to the PVPs. So the effect of beta-lactam is nullified. The third mechanism is efflux pump. You can see here some efflux pump are also present in the outer membrane. So if beta-lactam antibiotics enter through the influx pump and reach into the periplasmic space or inside the pathogen, the efflux pump antibiotic from inside the pathogen to outside, which lead to the neutralization of beta-lactam antibiotics. The most important mechanism which is developed by the microorganism is the hydrolyzing enzyme. So in this case, beta-lactam is an antibiotic, so the hydrolyzing enzyme is beta-lactamases, which is present in the periplasmic space. So once the beta-lactam antibiotic enter into the periplasmic space, it is cleaved by the beta-lactamases, hence the effect is nullified. So in this figure you can visualize the location of outer membrane, inner membrane proteins, periplasmic space protein, which include change in the influx pump, alteration in the target protein, expression of hydrolyzing enzyme, and overexpression or emergence of efflux pump. So for these four mechanisms, you can visualize it into the plasma membrane of bacteria. So bacteria can acquire resistance by any of the one mechanism which is described earlier, or by combination of multiple mechanisms. So this way, the bacteria acquire resistance against a wide spectrum of drug which by themselves differ in their mode of action. Such multi-drug resistant bacteria are of great concern because of unavailability of effective drugs against this resistant bacteria. The enzymes which are involved in drug resistance against the beta-lactams. The first enzyme which is involved is alteration in the penicillin binding protein. Although this penicillin binding protein is a target of this antibiotic, so beta-lactam enter into the cell and inhibit the penicillin binding protein. But the pathogen alter this penicillin binding protein and created resistance. The second proteins is efflux pump. They are involved in efflux out of antibiotic from inside of the bacterium to outside. The third enzymes are beta-lactamase enzyme, which hydrolyzing the beta-lactam. They are either metallo-beta-lactamases or serine type of beta-lactamases. And fourth one is alteration in outer membrane proteins, which is involved in the influx of the antibiotic. And the fifth mechanism, which is under research, is elevated mechanism of their metabolism, is also important in drug resistance. But these top four mechanisms are very significant to develop drug resistance against beta-lactam by microorganism. The penicillin binding proteins. The penicillin binding proteins are the enzymes that are required for biosynthesis of the cell wall. They are found in inner membrane of bacteria. So these inner membrane protein catalyzes final step of the polymerization, that is transglycosylation, and cross-linking, that is transpeptidation of peptidoglycan. So peptidoglycan is an essential component of bacterial cell wall. These inner membrane protein are the target of beta-lactam antibiotics. Beta-lactams are chemically similar to the substrate of these PVPs. Hence, they can be considered as a competitive inhibitor for PVPs. So they bind to the PVPs and block them, thereby disrupting the cross-linking and the formation of peptidoglycan layer. The resistance to beta-lactam antibiotic in some species has occurred by the development of altered PVPs with reduced affinity for antibiotics. These PVPs, that you can say that these modified PVPs can still perform their natural function but does not bind to the beta-lactam antibiotics. So development of PVP mediated resistance is thought to be a gradual process involving the introduction of multiple mutations in the PVPs. PVPs are divided into two subgroups. The first one is low molecular mass PVPs and second is high molecular mass PVPs enzyme. The high molecular mass enzyme are further divided into bifunctional class A enzyme, which possesses a beta-lactam insensitive transglycosylase domain and traditional beta-lactam sensitive transpeptidase domain, and the monofunctional transpeptidase class B enzyme. So we have bifunctional class A enzyme and monofunctional transpeptidase class B enzyme. The soluble low molecular mass PVPs has no identified role in beta-lactam resistance and are primarily involved in the carboxypeptidase reaction and peptidoglycan trimming. Despite their low sequence identity with more essential high molecular weight counterparts, low molecular mass PVPs has been common vehicle for inhibitor studies owing to their soluble nature. There are several PVP mediated mechanisms of beta-lactam resistance, including acquisition of new less sensitive enzyme, mutation of endogenous PVPs to lessen the reaction of beta-lactam, while maintaining some transpeptidase activity or by upregulation of PBP expression. The latter option to be the least effective, although the converse, that is lower expression of some resistant PBPs, might have proven to be useful to control the beta-lactam resistant bacteria. The second mechanism is efflux pumps. So efflux pump, which is one of the best characterized efflux pump, is max AB OPRM complex, and it is reported from opportunistic pathogen, that is Pseudomonas aeruginosa. So when you are talking about the efflux pumps, they are having three components. One component is present on the outer membrane, the other component is present in the periplasmic space, and third component is present into the inner membrane part. So this is the reason why they can efflux it, the antibiotic, from interior of the bacterium to outside. So this tripartite complex consists of inner membrane RND transporter max B, outer membrane porine, that is OPRM, and soluble periplasmic component, that is max A. Act on wide range of the antibiotics such as tetracycline, chloramphenicol, quinols, novobiacin, microlide, as well as beta-lactams. Not only that, they are also acting on the beta-lactamase inhibitors such as clavulanic acid. Other efflux pump such as ADE ABC efflux pump complex belongs to resistant nodulation cell division family, has been reported and characterized in few gram-negative bacteria such as Acinetobacter baumannii. Beta-lactamase enzyme. So beta-lactamase enzyme are periplasmic enzyme found in the gram-negative bacteria and assumed to be evolved from penicillin binding protein, which is a target of this antibiotics. It has been found that antimicrobial effect of the beta-lactam decreases due to the presence of beta-lactamases which hydrolyzing these antibiotics. These enzymes, these beta-lactamase enzyme, are expressed in high copy number. Copy number means number of beta-lactamases in drug resistance pathogen, and it is also found that it is induced by the beta-lactam, which is a molecule which undergoes cleavage by this particular enzyme. So this is reported that the expression of beta-lactamases in E. coli, Pseudomonas, Klebsiella and Acinetobacter and other pathogen leads to the development of resistance against this beta-lactam. Beta-lactamases are a group of enzyme which is classified into two groups, two major groups. The first group is chromosomal mediated beta-lactamases and second group is plasmid mediated beta-lactamases. There are more than 470 beta-lactamases which are identified till date, and they are classified into the four classes on the basis of their sequence similarity. So we have class A, class B, class C, class D. Class A, C and D are serine based class and class B are metallo based class. So in this way you can say that beta-lactamases is either classified into serine and metallo beta-lactamases depend on their mode of action, or you can say molecular classification, or you can say that they have four classes, class A, class B, class C, on the basis of their function or functional classification. The first class of beta-lactamase are serine type of beta-lactamases, and this is comprising class A beta-lactamases, class C beta-lactamases and class D beta-lactamases. Different group of these beta-lactamases are inhibited by different inhibitors which are used to differentiate one class of the serine beta-lactamases from other class. First of all, class A beta-lactamases. These beta-lactamases are inhibited by clavulanic acid and oxacillin, but not by azoterenum and EDTA. And this is a characteristic test for them. And they include TEM, such as TEM12 of E. coli, which is having 29 kilodalton molecular weight, SHV, such as SHV1 of E. coli, which have molecular weight of 28.8 kilodalton. The second group of serine type of beta-lactamases are class C beta-lactamases. This type of beta-lactamases are not inhibited by clavulanic acid. If you remember, class A, they are inhibited by clavulanic acid. So, class C beta-lactamases are not inhibited by clavulanic acid, but inhibited by oxacillin and azoterenum. If you remember, class A is not inhibited by azoterenum. So, this example of class C beta-lactamases include chromosomal MC of E. coli. Third group of this serine type of beta-lactamases are class B beta-lactamases. These beta-lactamases are poorly inhibited by clavulanic acid, but not by oxacillin. They hydrolyzing oxacillin faster than any other group of beta-lactamases, and they include oxa beta-lactamases such as oxa1 of E. coli, oxa24 of Acinetobacter baumannii, oxa51 of Acinetobacter baumannii. In gram-negative bacteria such as Klebsiella, Pseudomonas, Proteus, others, the presence and absence of beta-lactamases has been demonstrated. The sequence analysis of some beta-lactamases has been done, and it shows that there is some similarity in the sequence of these organisms. The widespread use of carbapenem, monobactam, and other antibiotics in past few decades has led to the evolution of new generation of beta-lactamases which have extended substrate spectrum, that is extended spectrum beta-lactamases or ESBL, as well as the development of novel carbapenemases and plasmid mediated MPC beta-lactamases and oxa beta-lactamases. So common ESBL variant include class A beta-lactamases such as TEM, SHV, CTXM, and other class D beta-lactamases such as OXA. These enzymes are classified by a broad substrate spectrum which include cephalosporin, azotenum, and other substrate. In this table, we have summarized all the beta-lactamases with their specific substrate, their inhibitors, and their representative enzyme. So, different classes of beta-lactamase as per molecular classification given by Ambler, the class A beta-lactamases, class C beta-lactamases, class D beta-lactamases, undefined beta-lactamases, and then metallo-beta-lactamases, that is class B beta-lactamases. Bush et al. Also classified the enzyme, and their number start from 1, 2, 3 and 4, and 2 is further divided in 2A, 2B and other classes. So class A beta-lactamase, if you compare Ambler and Bush, you will find that class A beta-lactamase of Ambler are further divided into some classes of Bush et al. And different classes of Bush et al. Are having a specific substrate. For example, 2A have the substrate penicillin and they have different inhibitor. So for example, 2A class of beta-lactamases have the substrate penicillin but inhibited by clavulanic acid, not by azoterenum and EDTA. Similarly, if you see that class C beta-lactamases, that is a class 1 of Bush et al. That is inhibited by azoterenum, not by clavulanic acid and EDTA, and their example is MC enzyme. So this table summarizes all the classes, their substrate and their inhibitor and representative enzyme of beta-lactamases. Serine type of beta-lactamases structurally have similar folds and their mechanism are quite similar, and their mechanism involve creation of a serine nucleophile by deprotonation of active site serine with the general base. Second point is The nucleophilic attack by beta-lactam ring to form a acyl enzyme intermediate, and the third one is the hydrolysis of the intermediate using general base activated water molecule. They have common three elements 1, 2, 3 in their active site. So if you see class A, class C and class D, they have similarity in mechanism as well as their catalytic element present in the active site. The other class of beta-lactamase are metallo-beta-lactamases or class B beta-lactamases. These beta-lactamases are poorly inhibited by clavulanic acid but inhibited by EDTA. And these beta-lactamases includes IMP and WIMP. A NDM beta-lactamase, that is New Delhi metallolactamase, is an emerging as an alarming rate and belongs to this group of beta-lactamases. Structurally, metallo-beta-lactamases and serine beta-lactamases are different. By sequence analysis, characteristic motifs of beta-lactamases are detected, which is also present in other protein and making this metallo-beta-lactamase as a superfamily. So class B beta-lactamases contains metal such as zinc and other. So it can be a zinc metallolactamase for a few metallo-beta-lactamases, and it is completely distinct from serine beta-lactamases in their sequence, fold and mechanism. There are three subclasses of class B metallo-beta-lactamases, which is known as B1, B2 and B3. Three classes of metallo-beta-lactamases, class B1 and B3 can bind to 1 or 2 zinc ion, whereas class B2 appears to be mononuclear. In the binuclear metallo-beta-lactamases, that is from B1 and B3, the zinc ions are proximal to each other and separated by a bridging hydroxide that has been proven to be attacking nucleophile in beta-lactam hydrolysis. The class B1 and B3 metallolactamases can also function as mononuclear enzyme in which single zinc ion that occupies the Z1 site coordinate the nucleophilic hydroxides. This mechanism has been proposed to predominate the presence of substrate under physiological conditions. The high reactivity of metalloenzyme is due to the presence of zinc-bound water molecules. Here in the table you can see the different characteristics of beta-lactamases and their active site elements. Here in the table you can see the class A have the secondary structure, that is 44% alpha helix, 17% beta sheet and 39% of the beta turn. Although this is explained for a given reference, that is TAM1, similarly for other classes we also have the difference in the secondary structure. The most important point here is the element 1, 2 and 3. They are important for catalysis by serine beta-lactamases. As you can see that the element 1 have serine residue in all the A, B, C beta-lactamases playing a role in nucleophilic reaction. In element 2, serine residue has been suggested to have a role in proton transfer during opening of the beta-lactam, or you can say the cleavage of the beta-lactam, and may act as a nucleophile in secondary reaction with some beta-lactamases. Tyrosine has been suggested to act as a general base in both acylation and deacylation or only in the protonation of desilation reaction. So this is the probable role that is played by serine and tyrosine. And in the element C, if you see, the lysine has been proposed to involve in acylation of element 2 and serine or tyrosine and serine, threonine or serine in substrate recognition. The common mechanism of all active site of serine beta-lactamases and PBPs is that they undergoes acylation with an important active site serine residues, and serine and lysine residues from element 1 and glycine residue from element 3 will appear to be minimum requisite for acylation step for both beta-lactamases and PBPs. However, it is a deacylation behavior of the beta-lactamases which make it mechanically different from PVPs, as in the class A beta-lactamases, glutamine play a role of activating the water molecule in the deacylation of acyl enzyme intermediate. In the mechanism, you can see that beta-lactamase interact with the substrate with a fast acylation step, and slow deacylation step result into the release of the product. After discussion of role of different proteins or enzyme in the drug resistance, let's have discuss what is the future prospects of enzyme of the drug resistance. So with the development of different techniques such as phosphoproteomics, immunoproteomics, the study of drug resistance significantly increased, and these methods have emerged to understand virulence and pathogenesis of resistant microbes. Literatures based on the proteomics method have shows that presence of differentially expressed protein in the resistance strain, but their actual roles in the resistance have not been confirmed using knockout or auxotrop from resistant factor. So if you want to signify the role of different proteins identified by the proteomics, then we have to create the knockout or we have to create the auxotrop of the resistant factors. Therefore, quantitative proteomics, which include above explained proteomics, are also requires the support from non-proteomic methods like auxotrop generation and other. Hence, we can say that with the development of quantitative immunoproteomics, quantitative phosphoproteomics and other omics method, the identification of different expressed resistant factor become more reliable. So we have to focus on different approaches to validate the role of particular protein in the drug resistance and design a specific drug against the identified enzymes involved in drug resistance. So let's summarize what we have discussed in this module. So in this module, we have discussed about different resistance mechanism adopted by gram-negative bacteria to overcome different antibiotics with reference to beta-lactam antibiotics, because we have discussed beta-lactam resistance as a model. In this module, we have discussed how different pathways or proteins which are mainly involved in the normal action of antibiotics are utilized by the pathogen and altered by the pathogen to control the effect of that antibiotics. We have discussed how the different resistance mechanism against beta-lactam antibiotics has emerged. The first mechanism is target modification. So the resistance pathogen modifies the PVPs, which is a target protein of this particular enzyme, which is target protein for this particular antibiotic. The second mechanism is efflux pump. So efflux pumps are emerged in the resistant strain to overcome the effect of antibiotics, because efflux pump pumps out the antibiotics. The third mechanism which is developed by the resistant strain is change in the permeability of the outer membrane. So the influx pump modified by the resistant strain basically inhibits the antibiotic to enter into the pathogen. And the most advanced mechanism which is developed by the pathogen is the emergence of hydrolyzing enzyme or beta-lactamases in this case. So beta-lactamases cleaves the beta-lactam antibiotics. Recently a new mechanism is also emerged, and that is the elevated metabolism. But this is under the research, how this elevated metabolism is affecting the resistance of pathogens. So different microbes either in alone or in combination of the mechanism, they inactivate the beta lactam and other antibiotics. So you can say that the different mechanism developed by the pathogenic bacteria are important for mankind, because they are the one which influence the dose of antibiotic which is taken by human being, and they also influence the efficacy of antibiotics. So the protein and enzyme which are involved in the drug resistance is important to study, because we can manipulate these enzymes, we can manipulate the antibiotics to overcome the effect of drug resistance.